Imagine you are a microbiologist facing an invisible army of bacteria. Some have thick armor while others have very minimum shield. How do you tell them apart under a microscope? The answer lies in the technique we are going to study today in microbiology is gram staining. Today we'll uncover the architecture of bacteria and their cell wall and discover why some bacteria appears to be purple and while others appears to be pink under the microscope when stained with gram stain. So the prokaryotic cells, before understanding the gram staining we need to understand the bacterial architecture. So prokaryotic cells have outer cell wall which gives protection to the bacteria. This wall gives bacteria their shape and prevents them from bursting in the harsh environment. Think of it as a microscopic fortress protecting the cell from the outside world. It also helps in surviving the osmotic stress. Imagine placing bacteria in a pure water and water rushes into the cell due to osmosis. Without the cell wall, the bacteria would swell and burst. But with wall, the wall acts as a armor and resisting the internal pressure, keeping the bacteria cell intact. In a concentrated environment, it also happens prevent excessive water loss, which is known as plasmolysis. So now what is the building materials of cell wall in different organisms? In case of plants, it is made up of cellulose or chitin. In case of archaeobacteria, it is made up of polysaccharide and protein, which doesn't have peptidoglycan. But in case of bacteria, it is made up of peptidoglycan. So this molecule, the peptidoglycan, is found extensively in bacteria and will be used as a tool to stain the bacteria and see. So meet the peptidoglycan. This is uniquely present in bacterial cell wall and it is a polymer which is highly durable. So these are repeating sugar units which run horizontally and many chains run parallelly, and these sugar units are connected by short peptide chain to form a unbreakable and multi-layered structure. This thick layer is key in gram staining. The story started in 19th century when Hans Christian Gran developed a technique to use this cell wall difference between bacteria and classify them based on the stain it appeared in the microscope after gram staining. So all the process is divided into 4 steps. We'll see later. First step is staining. So what happens in staining? Staining is primarily done with crystal violet. This, being positively charged, binds to negatively charged components of the cell wall, trapping the color of crystal violet. Next comes the iodine, which act as a mordant. Iodine forms large and insoluble complex with crystal violet, which fixes the color of crystal violet to the cell wall. Then happens the ethanol, which act as a decolorizer. So since in case of some bacteria which we say as gram positive bacteria, they retain the dye due to their thick peptidoglycan layer, but in case of gram negative bacteria they lose the dye entirely since their peptidoglycan layer is very thin. Next comes the counterstain, which is safranin. It act as a secondary dye. Now in case of gram positive bacteria which appears to be purple, as stained with crystal violet, it masks the color of safranin, which is lighter in color. But in case of gram negative bacteria, since the crystal violet color was removed by ethanol, now new color which is of safranin appears in gram negative bacteria. So how does this happen? So in case of gram positive bacteria, the thick peptidoglycan layer, this single dense wall is the reason for the crystal violet and iodine complex to be trapped inside, which is giving purple color to the bacteria. But in case of gram negative bacteria, since it is thin and the peptidoglycan layer is also thin in this, so it fails to trap the purple color of crystal violet. Thus it is easily washed off with the ethanol and stained by the counterstain, which here is Safranin. So this is the ultimate difference between gram positive and gram negative bacteria. So gram positive bacteria's peptidoglycan layer is thick, while gram negative have thin layer. The outer membrane of gram positive is absent, whereas gram negative it is present. LPS is absent in gram positive bacteria, but in case of gram negative bacteria it is present and kind of gives shield to the bacteria. So when seen under the microscope, the final color observed for the gram positive bacteria is purple, whereas gram negative bacteria is a pink or red type. So how do we use this gram staining as a tool in medical study? So what happens is when any medical doctors observe the color of bacteria under the microscope, they kind of look for the structure and prescribe the medicine for the whether it is gram positive and gram negative bacteria, so that the disease is cured for the patient. Thank you.